To kick off the fantastic program this year, we're about to hear from Professor Adrian Hill from the University of Oxford. Adrian is the Lakshmi Mittal Professor of Vaccinology and Director of the Jenner Institute at Oxford University. In 2005, he founded the Jenner Institute at Oxford, which is now one of the largest academic vaccine centres globally with clinical stage vaccine programs against 15 diseases. Please join me in extending a warm welcome to Adrian, whose talk is a high-efficacy malaria vaccine. Adrian.
Ladies and gentlemen, Professor Bezra, thank you very much for inviting me, and congratulations on your timing. None of you could have known that this lecture would be taking place within less than a week of the first malaria vaccine ever being licensed by any country. So I'm here to tell you a little bit about that story today, and not too much, I hope, about the last 30 years of struggling we've had trying to make malaria vaccines. But it is an honor to be here. Fantastic to see so many people at this society. And let me delve into malaria. First of all, disclosure of interests. As you'll hear, I've been working fairly closely with the Serum Institute of India over the last few years, and I'm a co-founder of two small biotechs, neither of which I've persuaded to work on malaria. Now, malaria really is a problem, and has been for, well, we don't really know how long, but if you can do phylogenies and root the origin of diversity in malaria parasites, it goes back about 30 million years. So we've co-evolved with malaria. And of course, we weren't around 30 million years ago as human beings, so we've speciated with malaria. And the point really is this parasite's had a long time to think about our immune system and how to survive, and has done pretty well from a parasite's point of view. Its effects, of course, are imprinted on our genome. Everyone knows about the sickle hemoglobin mutation being pretty strongly protective against clinical malaria. There are now lots of other loci from GWAS studies and so on, showing just how important our need to adapt to malaria has been and how it's literally changed us. Now, of course, malaria used to be far more widespread than it is today. That's the good news. In exotic places like Cambridge, the swamps of Washington, D.C., famously, of course, in the Mediterranean, and the history books are full of stories about famous people you'll have heard of who probably died of malaria. No parasitological diagnosis. But of course, over the last 30, 40 years, it has been retreating towards tropical regions. But it's by no means finished yet. Progress, to be frank, in controlling malaria has stalled. In about 2000, there was a lot of interest. It became a front-page news story that we and Bono and Bob Geldof and everybody were going to do something about deprivation and poverty, and particularly disease in the developing world. And new money became available for global health, and particularly for malaria. Well, the bad news is that the number of malaria cases is about the same today as it was in 2000. Malaria deaths have fallen. It was about 800,000 to 1 million. Difficult to estimate those numbers in 20 years ago. And today it's down to about 640,000. And the WHO and other eminent organizations have detailed plans to eradicate malaria or to eliminate it as a global health problem by, say, 2030. They don't look totally convincing, and certainly won't be unless we have new tools. So, as you know, malaria mainly affects children, mainly kills children, because you develop immunity as you grow up, as a young child and then an older child, in areas where there's a lot of malaria, and that naturally acquired immunity, by and large, stops you dying from malaria after the age of 6 or 7, unless you become pregnant. And malaria in pregnancy is a distinct problem. The parasite likes living in the placenta, and this causes stillbirths and other problems and can kill the mother as well. So in numbers, there are around about 250 million clinical episodes of malaria. Some of my colleagues call that mild malaria. If you've had an episode of malaria, I don't think you'll call it mild. And 84 countries in the world still have malaria as an endemic disease, mainly in Africa, so it's mainly an African problem with most of the deaths there. But this is falciparum malaria that kills you. I'll come to vivax malaria, the other parasite that's actually more widespread, South America, Melanesia, and everywhere in between, towards the end. But that's less of a public health problem because of its minimal mortality. So what are we doing? We're doing quite a lot as a global community in controlling this. $4 billion was spent last year and the year before on malaria control, using tools that were around literally decades ago. Spraying insecticides, using bed nets, using drugs. We actually give drugs monthly to children in highly endemic areas. The problem with the drugs is they last days to weeks, not months to years. So you have to keep giving them, and logistically that's quite difficult, but they do work. And the punchline here on that slide is there is no deployed vaccine. That's still true today. What happened last week was we had the first nationally licensed vaccine. And of course, right through the COVID pandemic, those of us who work on malaria were fully aware that at least in Africa, there were more people, mainly children, dying of malaria than dying of COVID. And I've met many people from Africa who said, what was all this fuss about you guys in rich countries worried about COVID? Why don't you do something about our malaria problem? So, people have been trying. This is my way of saying it hasn't been an easy vaccine to make. These are two brothers, the Sargent brothers, working in Algiers in the early years of the last century. That's the first paper I can find on malaria vaccines, 1910. And they were attenuating parasites in the way that Pasteur had taught us. And they were trying to make a malaria vaccine that did have some partial efficacy in their preclinical model, but that was actually bird malaria. So not very close to the action that we needed for a human malaria vaccine. I pulled this off the Financial Times website. It really makes the point, I think, even more strongly. And what I like is that the two things the Jenner Institute has been mainly involved with in the last 5 years has been the top left here and the bottom right. The bottom right was, as you know, the fastest vaccine development program in history on COVID-19. On the top left, we've also been involved in the slowest, which is malaria, going back to 1880 with the discovery of the infectious cause of malaria, and now 2023, after 145 years, we have a vaccine that I hope will be useful, and lots of things in between. So why is it so difficult? Well, one of the problems is that if you're going to induce antibodies by your vaccine, you need levels of antibodies that are probably a log, if not 2 logs, higher than you get with most vaccines. And those are vaccines that work. So malaria is uniquely difficult. That's probably because the parasite's been evolving, as I mentioned earlier, to evade the immune system. If it didn't do this, it wouldn't be around still. And then there's another opportunity, I'll come to in a moment, where you can use T-cell immunity to protect against malaria. We can actually do that. It actually works, just not well enough. But again, the numbers of T-cells you need are really very high indeed. So here are our excuses for not having a malaria vaccine until now. 5,000 genes. In your virus, there will be fewer, a lot fewer, 100 times less than, say, in a coronavirus. Substantial stage specificity of antigen expression. So the good news is there are 4 stages to the life cycle. If you make a vaccine against any of those 4 stages that works really well, you're probably there. But they're different, those vaccines, because different stages of the life cycle express different antigens. I'll come to the cartoon of the life cycle in a moment. Antigenic variation, famously like in trypanosomes, malaria has antigens that switch on and off within days so it can evade blood stage immunity. Antigenic polymorphism accumulated over millions of years is there particularly at the blood stage, less at other stages. And it's very clear that the parasite has evolved to subvert and evade host immunity. I could give you very detailed lectures on how it does that and what it's evolved to make work, but we'll skip that today. But the phrase we like to use is unnatural immunity. We're trying to make immune responses that you never see in nature. Even in people who've been exposed to malaria for decades and decades and decades and have strong immunity, their levels of antibodies are nothing close to what we can induce by vaccination today, at least against single antigens. So natural immunity to malaria is a compendium of weakish immune responses against lots of different blood stage antigens. So let's remind you of the malaria parasite life cycle. It starts, of course, with a mosquito bite inoculating sporozoites. Modest, frustratingly small numbers, maybe 20 or 30 go into your arm. There may be some immunity in the skin, not very much, but those parasites quickly go to your liver within maybe half an hour. Once they're in the liver, they're hidden from antibodies. They are multiplying like crazy. I mean, one parasite goes into a liver cell, 20,000 come out 7 days later. Then the blood stage is when you might begin to feel unwell. We know a lot about this because we infect a lot of people. I'll come to that in a moment. And you don't get symptoms until 4 or 5 days after you've had blood stage infection because the parasites need to multiply, maybe tenfold every 48 hours. And that's when you start feeling seriously unwell. Parasites start sequestering in important tissue beds like in your brain, and you may end up with cerebral malaria and die from that. Or if you control the malaria a bit better, then you'll lose lots of red cells and become anemic and might die of severe anemia. And then something really curious happens. The parasite changes again into a different mode or different stage of the life cycle, which are these gametes or gametocytes inside or outside of red cells, which are taken up by a further mosquito bite here, and the life cycle continues with sex occurring bizarrely in the midgut of the mosquito, creating new sporozoites. So people have tried every one of these stages to make vaccines against them. They've all been difficult, and to spoil the story, it's antisporozoite vaccines that have been most successful. Possibly because the number of sporozoites being targeted is quite small compared to, say, blood stage vaccines. So this is a non-random selection of candidate vaccines that have been taken into clinical trials in people. The last time I counted, there were 143 different candidate vaccines that had been tested in the clinic. In other words, somebody had made them to GMP, spent millions of pounds doing that, and started a clinical trial. Now, you haven't heard of most of those because effectively 2 have been useful. What stages have they targeted? Well, the 2 that have been useful are against the sporozoite. I'm going to be talking about R21 for the rest of this talk. And you can see that's a nanoparticle or virus-like particle, about 22 nanometers in diameter. And our vaccine is coated with malaria on the surface of a hepatitis B vaccine, which you've almost certainly have had. That's at the sporozoite stage, so antibodies are involved here to stop liver infection. At the liver stage, you need T cells because your parasites are inside the liver cell, particularly CD8 T cells, which in mouse models can do a really good job and protect mice against malaria. You need viral vectors to get exceptionally strong CD8 T cells. This is the best way of inducing cellular immunity, not mRNA vaccines. That's very clear from the COVID experience, and they've been partially successful. The earliest vaccines were against the blood stage on the simple argument that it's the blood stage that kills you. Let's make vaccines against the stage that's a problem. That's been super difficult. There's now one antigen that unusually for this stage is highly conserved. It's called Rh5. Maybe there are a couple of others that are involved in entry into the red cell. And my colleague Simon Draper in Oxford has the world's leading blood stage vaccine candidate, Rh5, coming along very quickly. And finally, To block transmission and eradicate malaria, you very likely need a vaccine against the transmission stages, against the gametocytes or even the zygotes inside the mosquito, and that's a completely different immunogen that you would need to stop transmission inside mosquitoes and prevent them infecting anybody else. And all of those efforts have been ongoing for decades now. But let's cut to the chase, to the two vaccines that have shown significant, and I would argue useful, efficacy in humans. The first one was not us, it was RTSS from GSK, way back when they were SmithKline-Beecham in Rixensart in Belgium, making candidate vaccines for malaria in collaboration with the US military, who really wanted a vaccine for deployment in certain regions of the world. In about 1984, it was discovered that the main coat protein of those sporozoites is CSP, the circumsporozoite protein, that very usefully has a central repeat of 20 to 40 copies of just 4 amino acids, NAMP. And the optimists said, wow, that's easy. We'll have a vaccine in a few years. Didn't quite work out like that. But there's the whole protein. There's what's in RTSS. R stands for the repeat, T for the T-cell region in the C terminus, and then fused to the hepatitis B surface antigen. This was a very clever trick invented way back in the 1980s by Joe Cohen in Rixensart when SmithKline-Beecham were first working on malaria. And by multimerizing that protein into a nanoparticle with the hepatitis B as the core, you got much stronger antibody responses. And that's why this vaccine was the first to show significant protection in human beings back in the 1990s. To do that, Joe had to make an excess of hepatitis B in the yeast cell that was producing the vaccine. This then multimerized or made a nanoparticle, which I'll show you in a moment. A good 20 years later, Catherine Collins, a graduate student in my lab, managed to express the fusion protein without the excess of hepatitis B surface antigen. A different yeast strain, a stronger promoter in that yeast, Hansenula rather than Saccharomyces for those interested. And you have a particle now where all of the molecules encode CS. And this turns out to be important. There's CAS. These are the cartoons of the two nanoparticles. There's RTSS with little islands of the malaria antigen, the circumsporozoite protein on its surface. And there's R21, where every Hep B molecule in blue here has the malaria protein fused to it. And very briefly, to spoil the story again, the efficacy of RTSS has been significant and worthwhile, but not very high. It does use an extremely potent adjuvant. Some of you will have heard of AS01. It's in the shingles vaccine from GSK. And we managed to get higher efficacy that I'll tell you about in a moment with this, we think, improved immunogen, using a simpler to manufacture adjuvant called Matrix M, which was in the Novavax COVID-19 vaccine. So very briefly, what I'll do is run you through the 4 clinical trials that we've done with R21, a challenge trial in the UK, VAC72. You can see we were doing quite a few trials before we got to R21. VAC73 in coastal Kenya, an age de-escalation study in Kenyan adults down to infants, and 2 critical efficacy trials, VAC76, a phase 2 trial in Burkina Faso, and VAC78, which has not been published yet, but I'll show you the recent data. That's our phase 3 trial. So, how do we screen malaria vaccines once we've shown some success in mice? Well, the general way of doing that is to do what we would call a challenge study, or a controlled human malaria infection with these friends, which are our Anopheles mosquitoes. They're hungry, we starve them overnight. They've been fed on infected blood, so they have parasites in their salivary glands, and people like, well, you, rather than me, will go and put their hands on these little coffee cups. They have to be espresso, not cappuccino, those are too big, with some gauze to stop the investigators getting infected. And this nearly always works. So if you challenge, as we say, dozens of people, about 95% of them will be infected if you get 5 mosquito bites. We do this at Imperial College London, which is one of the best insectaries in the country, and have been doing this with us for a long time. We've done over 20 challenge studies in all to develop vaccines. And what we found for the first time with this vaccine was that suddenly we got 65 to 82% efficacy. In other words, most of the volunteers were protected for the first time, albeit in small numbers. We don't challenge hundreds of people, we do tens. Nonetheless, that was really exciting. The immune responses were unusually potent. So we moved on to a trial in Kenya. There is an Oxford unit on the Kenyan coast in Kilifi, funded by the Wellcome Trust, and they've done lots of malaria trials, and they did this one in 92 adults, children, and infants. You start in the adults for ethical reasons, and then age de-escalate into infants, and that was safe. I won't show you that, but the immune responses, happily, were much higher than in UK adults. 12 months of age seems to be the age when you make the best immune response to many foreign antigens. Then we moved on to the third trial, which really got us excited, in Noro, Burkina Faso. So why did we go to a country that many people have almost never heard of in West Africa? Well, very simply because places like Noro, an hour and a half's drive from the capital Ouagadougou, have ridiculously high levels of malaria. It's seasonal. There's a burst of transmission after the rains in August. 8 episodes would not be really unusual. We've seen children with that in a single malaria season. So a lot of power in the clinical trial to detect whether the vaccine is working or not. We've also worked down here in the southwest where there's less seasonal transmission, but the key reason we went is there are fantastic investigators here where, of course, malaria is the biggest health problem in the country. There's the rainy season, July to November, really peaking in September, October, huge rainfall, and great seasonality, so by February, March, April, there's far less malaria. What we did was to take the R21 vaccine. We found we needed to give 3 doses to get maximal immunogenicity. We didn't know the best dose of adjuvant. The adjuvant here is important. It's called matrix M. It's a saponin adjuvant, which enhances the antibody response. 50 micrograms, as I'll show you, turns out to be better than 25 micrograms. And then you have a control group of children who were, in this case, given a rabies vaccine. So 3 doses, and then followed through that really high transmission malaria season. Starting back in 2019, we're now up to almost 4 years of follow-up. And what we saw was very exciting. So what you're looking at here is the cumulative incidence of malaria over days. So that's a year here, just beyond 300 days. And in the control group, you have a lot of malaria, most of the children getting infected. There's the malaria season. It flattens out in the dry season. And here's what we saw with our vaccine. And the numbers are over here. With the lower dose of adjuvant, 69% efficacy. With the higher dose, 77% efficacy, which was completely unprecedented in the field and highly statistically significant. So that was published just about 2 years ago. And that was better than the only other candidate really in the field that was being developed by GSK. That's the RTSS that I showed you a couple of minutes ago. They had also done a trial at multiple sites in Africa, but particularly in Noro, where the efficacy was 44%. So we were encouraged that we were seeing significantly higher efficacy, partly because of a better immunization timing, but partly because the vaccine itself is more effective. So that allowed us to bring in an industrial partner. Amazingly, they found us. We go around the world talking to companies trying to interest them in malaria. Umesh Shaligram from the Serum Institute of India turned up in my office saying, "We hear you have interesting data." So they funded this phase 3 trial, and you're now into tens of millions of dollars, not millions, to do these trials. We have 5 sites in Africa, in West Africa, in Burkina. We've mentioned Noro, but also in Mali, Bagamoyo in Tanzania, and Kilifi in Kenya again. Our statisticians are at the London School of Hygiene and Tropical Medicine, great malaria expertise there. Novavax provide the adjuvant, and the Serum Institute of India are the commercial developer. So we chose to go not just to Noro and Buguni, high transmission sites in West Africa, but to less seasonal malaria sites in East Africa, Kilifi and Bagamoyo, with much lower transmission, and Dande in Burkina Faso, which is in between, a lot more fairly high transmission, but much less seasonal. So we have now a 2-to-1 randomization of the favored dose of adjuvant. One, again, the controls receive rabies vaccine. The sample size is 10-fold higher, 4,800 children, because you need 3,000 children safely dosed for registration. With 2-to-1 randomization, you just get there. Double blind, of course. Other malaria interventions that are used routinely, like giving bed nets and insecticides, are given to all the children. So that's standard of care, if you like. The University in Oxford sponsored this, and the Serum Institute of India funded it. And the other change was, I may have omitted to mention that in the Noro trial, it was 5 to 17-month-olds, the children who are most at risk with little immunity. We extended it up to 36 months in this trial, partly to speed recruitment, but mainly to ask whether older children could be protected as well. So here are the groups. There's the regimen, 0, 28, and 56 days, and then a booster dose at a year, which we'd already seen reinvigorated the antibody responses. So, in brief, the punchline is here, and these data are not published. They hopefully will be soon, but at the seasonal sites, as in Noro, 75% vaccine efficacy, with confidence intervals that other vaccinologists working on different diseases look at with envy. These are really tight confidence interval. The lower bound is 72%. So the vaccine's working at least 70% in this population. In the standard sites where there's less seasonal transmission, 67%, not significantly different from the 75%. And those are the individual sites, a little bit lower in East Africa, but not significantly. So that's over a 1 year of follow-up, which was our primary trial endpoint, and that result came in late last year. Interestingly, if you look at the younger children and the older children, there is a difference that is just statistically significant, as shown here. This is comparing the 5 to 17-month-olds to the older children, and it's better in the younger children. In fact, it's 80%, which is a remarkably high figure. Their antibody titers, I won't show you the data, are higher as well, so we think this is real. And the good news, of course, is when you go vaccinating in deployment, you're going to start in infancy, probably at 5, 6, and 7 months of age, so that's probably the relevant figure. High in seasonal sites, and now particularly so at the standard non-seasonal sites. What about severe malaria? This is what you really want to prevent. Now, happily, if you go and do a phase 3 trial with all the staff that entail and the monitoring and so on, severe malarias and deaths particularly fall because you treat children earlier. But we did have enough severe malaria cases just to see a signal of 63% here. Now, that should be the same as the 75%. It certainly is within those wide confidence intervals, so we have no reason to believe that the vaccine will be any less or more effective against severe malaria than clinical malaria. So that's in the first year. What happens in the second year? And of course, you'd like as durable protection as you can. You don't need it to be lifelong, because once you're 3 or 4 or certainly 5 years of age, you're largely out of the woods as an African child. You've acquired enough natural immunity. So we only have 6 months more data from the seasonal sites at the moment. And again, in the second year, at least in the first 6 months, 75% efficacy, which is what you might expect. And similarly, at the standard sites, 73%. So that was all looking good. We looked at a lot of other potential confounders that might have an impact. I've mentioned already that there is higher immunogenicity in the very young children. There was no impact of having drugs at the same time against malaria. Seasonal malaria chemoprevention was independently additive to the vaccine. Even the very malnourished children that you occasionally see in these populations had good vaccine efficacy. And there was a trial pause that led to a third dose being given much later at 4 months, or up to 4 months. There was no impact on vaccine efficacy, and efficacy was the same in males and females. Serious adverse events is what you worry about most in a phase 3 trial, because if there is one or there's a safety problem, the vaccine isn't going anywhere. We saw essentially no signals. In summary, the only adverse event that seems to be possibly related to vaccine, and the numbers are small, are 5 febrile convulsions in the vaccinees compared to 1 in the controls, 2-to-1 randomization. That's nowhere near significant, but I suspect it's real because you do see that side effect with the adjuvant we're using. It's self-limiting. There's no long-term effect. Meanwhile, of course, that phase 2 trial in Burkina Faso was still ongoing, and we've accumulated data over a longer period of time. To be precise, nearly 4 years, well, 3.5 years on this slide. And the cumulative efficacy over time is pretty well maintained. This has been a big issue for the malaria field. Would your vaccine just be working for months or for a year and then wane completely? Some vaccines have had that issue because their immunogenicity is not as high. Here, this is pretty good, actually, 66% over 42 months. There's certainly no evidence that the efficacy is going negative over 3.5 years. And that's just to underscore this point. Some people were worried that at the seasonal sites, where there's a shorter transmission window, you'd get better efficacy than at the standard sites. We don't see that. Here's the decline in efficacy over the first year. There is a signal. It is going down a little bit, but not different at the seasonal and standard sites. They overlap, and it's still pretty well maintained out at 12 months. Going into a little bit more detail, and I apologize, this is a little complicated. What we're doing here is comparing the loss of efficacy with 2 different vaccines. Ours, the R21 vaccine, 74% in the first half of the year, 63% in the second. So, as I said, a reduction, but not significantly so. And in fact, in the younger children, it's higher overall, 82%, not 66%. And there's the RTSS data published from their phase 3 trial, where the efficacy is significantly lower and wanes faster. So we're reassured that we're likely to be doing better in terms of maintenance of immunity than any other vaccine. For the immunologists amongst you, I'll just give you a glimpse of the data on one slide, and this is actually from the phase 2 trial. After 3 doses, you get very high antibody levels. That's probably in the hundreds of micrograms per mil. You can see that you do need the 3rd dose to get up above 10,000. And of course, these antibodies wane over time, and that's over a year. They're back not to baseline, but relatively low, about a 90% drop over the year. You can boost them. They go up almost as high again. But interestingly, that's higher than that, and the trough here is well maintained. So we are seeing a reduction in antibody levels. That happens with every vaccine. The point is that the avidity of these antibodies seems to be much higher. We're getting a better quality of antibodies, so out here, even though the levels are lower, you're seeing more efficacy for a certain given antibody titer if you go into the data and analyze it carefully. So this is still a challenge. We'd like to have an infinitely durable vaccine, but at the moment this is looking very usable. So, almost there. Let's just look at a couple of final considerations. On deployability, the Serum Institute of India are really good at this. They are the main supplier of vaccines to Africa. They don't sell expensive vaccines to Africa. Nobody would buy them. They wouldn't be able to. So, there are over 5 million doses available now. These are the practice runs, if you like. There was a formulation where we had to mix the adjuvant with the vaccine at the bedside, so to speak, or in the clinic. That's been corrected. We now have a single vial formulation. The shelf life is 2 years in a fridge. And remarkably, if you take it out of a fridge and you have to go out to a distant village and vaccinate there, for 2 weeks, the vaccine is stable at 40 degrees centigrade, which is remarkable. The scale of supply can be hundreds of millions of doses every year, which should be transformational. RTSS are hoping to distribute 4 million doses this year, 6 million next year, 8 million the following year. That's really, really not enough for 40 million children who need a vaccine, and multi-dose vials are coming soon as well. So in summary, we've seen no significant safety concerns in over 12,000 doses given to young African children. We've followed up at seasonal sites and at standard sites and see substantial efficacy before the booster and after the booster at both sets of sites. The numbers are here. You've seen those up in the 70% range, and possibly even higher efficacy in the younger children vaccinated, say at 5 months of age. And the fact that the cost here of this vaccine is going to be low, we're talking about $3, $4, $5 a dose, not double that. Very high supply level I've just mentioned, and likely much improved cost effectiveness because of these two factors. This should be a very deployable vaccine. Just to finish with, some of you will have had this vaccine, most of you will know about it. So did the experience with malaria help us make a COVID vaccine even faster? And the short answer is yes. The chimpanzee adenovirus, the Chad virus vaccine platform that was used for COVID was developed first for malaria, first clinical trial back in 2007 in Oxford. We had safety data from over 10 trials, which made our regulators happy when we were rushing, and 7 of these were malaria Chad vaccines. The platform had benefited from years of support from the Gates Foundation and the Wellcome Trust, speeding rapid vector generation. We've now made over 1,000 adenoviral vectored vaccines in the last 5 years. Manufacturing scale-up turned out to be crucial if you're going to vaccinate the world, as we tried to, and for malaria, and particularly early on, and then rapidly applied to COVID-19 by Sandy Douglas and his team at the Jenner Institute, allowing very low-cost supply to developing countries. And malaria research, therefore, essentially enabled the Serum Institute partnership, and they were decoding with us the malaria trial in January 2020 and seeing high efficacy for the first time, and that increased their enthusiasm to license a vaccine from a bunch of academics in a university, which doesn't, of course, usually happen for global response. And then Serum really overperformed. 60% of the 3 billion doses of the Chad vaccine made during the pandemic there were 25 manufacturing sites. One of them, being Serum Institute, produced 60% or more of the doses. And just to give you a glimpse of that, this is the room in Pune in India where all of those doses were made. Yes, 10,000-liter bioreactors, 6 of them in a row, but extraordinarily productive. And this was some of the team I visited at the time. So what's left to do in malaria? Sadly, quite a lot. R21 is a vaccine to prevent disease and death in young African children. What you'd really like to do with malaria is get rid of it, as we're trying to do with polio. There will be elimination trials coming up with this vaccine. It's not going to be completely effective. We need an even better vaccine to do that, but this is an interesting exploratory study. The leading blood stage antigen, Rh5, we're combining already with R21 in a trial about to start in the Gambia with Simon's group. And a malaria elimination vaccine would have R21 and transmission blocking vaccines that I really haven't talked about. There are several antigens being assessed by Shumi Bishwa's group in Oxford, and very encouraging recent data of 74% efficacy with an NIH group working in Mali. So plenty to do there in combination. But of course, as I mentioned, there's also another parasite, Plasmodium vivax, which is more widespread, just not as lethal as Plasmodium falciparum. If you were in South America, this is probably the vaccine you'd like to have most. We have an vivax, RVX21, that is in manufacture at Serum Institute. Again, looks very good preclinically. Combining these two is the obvious thing to do for an elimination vaccine, and possibly, in the future, a vaccine for people like you who might like to go traveling where there's a lot of malaria. And adding a transmission-blocking vaccine against both species and blood stage antigens, different stages as well, will probably be the final formulation that will get rid of malaria and eliminate it and eradicate it, ideally in the next decade. I don't think it's going to be in this decade. So I'd like to stop and thank the many people who've been involved in Oxford, Professor Katie Ewer on immunology, Sophie Fernando Marina, our lead clinician, Alison Laurie, head of regulatory, the amazing team and teams at the Serum Institute of India, and perhaps most of all, those who did most of the work on the phase 3 trial in 4 different countries in Africa. Thank you for your attention.